name's Amy Reeve and I'm a researcher at Newcastle University, and I am interested to understand how changes in energy production affect the brain cells of the substantia nigra. I've been asked some questions by people affected by Parkinson's and I'm going to answer those.
How did you become involved in the area of Parkinson's research?
So I did my undergraduate degree at Edinburgh, and my degree was in neuroscience, so trying to understand how the brain works and changes that we see in various diseases. And then when I came to want to do my PhD, the PhD that I applied for here at Newcastle was interested in looking at Parkinson's disease, and that's sort of really where my specific interest in Parkinson's started.
As Parkinson's UK is funding your research, can you explain what the process was for applying for this, and were there people with Parkinson's involved?
So I sent in my application for the fellowship around about December last year, and then around about Easter time I got an email to say I've got an interview, and then I went down to the Parkinson's UK headquarters in London and had an interview with 4 reviewers, two of which were Parkinson's patients who were part of the lay review board, and then there were 2 scientists on the panel as well, and they interviewed me for about half an hour to an hour. And then about 2 weeks later I got the email to say that I got my fellowship and I was being funded.
How do you work out how much the research project is actually going to cost?
So we work it out based on the things that we need to perform the research. So for example, if we need to look at cells, we might need to use a microscope, and using the microscope takes time and costs money, and also the things that we use in order to be able to visualise the cells. So for example, some of my studies I light up the mitochondria within the cells by putting fluorescence onto them, and that costs money. So basically our research costs as much as the things that we need to use to perform the research costs. So we sit down and we work out exactly the kind of experiments that we want to perform, and then work out how much each individual component will cost for the duration of that experiment.
How long have researchers known that mitochondrial defects could actually be involved in Parkinson's?
So we've known that the mitochondrial defects are likely to be important since the early 1980s, and there was a very specific example of sort of serendipitous, I suppose, scientific research that came out. So there were a group of intravenous drug users in California who got a batch of drug that had been contaminated with something called MPTP, and a few weeks after they took this drug they developed symptoms of Parkinson's. And when we looked further into the mechanisms that had caused that, we found that the drug had very specifically stopped the mitochondria working, and that had led to their symptoms. But ever since then, the amount of knowledge that we have about the impact of mitochondrial defects in Parkinson's has been increasing. So we know, for example, a lot of the inherited forms are caused by mutations in genes within the mitochondria.
Can you tell us about what mitochondria are?
Mitochondria are really important for the majority of the cells in our body, and they're really small little structures that have 2 membranes, and they're found, like I say, within the majority of the cells in our body. But they're really important because they generate the majority of the energy that our cells need in order to be able to carry out their various functions. So whether or not that's a muscle cell sort of contracting, or a heart cell, or any cell in our body. But the most interesting thing for us in terms of looking at Parkinson's is that the brain cells are really heavily energy dependent. So they need a lot of energy in order to be able to maintain their electrical activity, but also to be able to communicate with each other. So if our mitochondria don't work properly and they're not producing enough energy to maintain these properties within the brain cells, then this is obviously going to be quite detrimental for the cell. And in particular, when we look at brain cells, if brain cells can't communicate with each other and they can't maintain their electrical activity, then there is evidence to show that they will die. So obviously, if there are changes going on in the mitochondria aren't producing energy, then the brain cells won't be able to work properly, and this is why we think perhaps they might die in Parkinson's. So my research really tries to understand exactly how changes in mitochondria, and therefore changes in energy production, do affect the survival of cells in people affected by Parkinson's.
Researchers in Plymouth are studying fission and fusion. Can you briefly tell us a little bit about what this is, and does this reflect, or is it relevant in your research?
So it's quite relevant actually. So the mitochondria, if you look them up in a textbook, they're always pictured as being sort of jelly bean-like structures, but actually they're very dynamic and they like to stick together, but they also break apart. So in a healthy cell, for example, we see that the mitochondria form a really nice network that completely fills the cell. However, if the cell's not very well or it's unhealthy, then this network tends to break apart. So when then mitochondria stick together, this is fusion, and when they break apart, it's fission. So part of my research is actually looking into the processes of fission and fusion within brain tissue itself, and just trying to see whether or not we see changes in response to different mitochondrial defects.
What inspired you to become a scientist?
I think it was probably my dad, actually. My dad was a science teacher, and even from a very early age we used to do sciencey things together. And I remember when I was about 5 or 6 years old, he bought me like a Fisher-Price microscope thing, and we just used to potter around and have a look at things under the microscope. And then my interest just maintained then throughout school and into university.
If your research gives positive results, how long would it take for drugs to become available?
So in the 3rd year of my research project, I'm going to work with a pharmaceutical company who have a large batch of drugs and different chemicals. And what we're going to do is to screen these for those that have a positive effect on the cells and help the cells to be able to respond to mitochondrial defects and to be able to survive. But a number of these drugs are actually drugs that have already been approved for human use. We're just trying to find new uses for them, or perhaps they've been found to be less effective as we originally thought for their original purpose. So we're going to screen all these and then find out those that have the same effects that we're seeing in patients with inherited defects. But in terms of how long it will take to get to a cure, I guess it's really hard to tell because it will depend on the results that we get out at the end of the fellowship.
What has been your career highlight, would you say?
Actually, probably getting my fellowship. The really good thing about the fellowship is that I get to obviously work with the charity and get involved in sort of some of the groups for people affected by Parkinson's, but also actually it's allowed me to become much more of an independent researcher, so it's allowing me to drive my research in the direction that I want to go in, and hopefully then be able to actually move my career forward as well.
The research you've conducted so far, have the findings been as you expected, or have any surprised you?
I've done a few studies where actually the results that we've got have been the complete opposite to what we thought. So, for example, a couple of years ago I did a study where we looked at the relationship between Lewy bodies and mitochondrial defects, and we thought that we would find Lewy bodies in cells that had a mitochondrial defect, but actually when we looked, the cells that had the Lewy bodies seemed to have an increased expression of some of the proteins that are really important for generating the energy within the mitochondria. So this was completely unexpected to us, but it has meant that our hypothesis and our ideas can develop over time, so it's really interesting and exciting.
How do you collaborate with other researchers, and do you share findings?
So I collaborate quite a lot with people in Newcastle, so they're really good. For example, if you've got some interesting results and you want to get an initial response. But I also collaborate with some researchers at UCL in London, and we work together very closely on a project. So, for example, I take my cells that I grow in the lab down there, and I can use all their expertise and their equipment and some of the techniques that they're really good at in order to be able to expand the research that we're doing. And we'll publish papers together and things like that. But it is really good to have an expert in a different field who's able to give you input into your research projects.
How important is it for you to hear from people with Parkinson's and those essentially who will hopefully benefit from your work? And do their opinions and thoughts ever guide the work that you do?
It's really important to me, actually. It's a really good motivator to be in contact with people affected by Parkinson's, to hear sort of what they're interested in and what they think people should be researching. And I really like to hear their questions and actually their opinions on my research. I think it's really helpful to see how your research fits in with what people are hoping for or are interested in. And I always really like answering people's questions and just hearing what they've got to say, basically hearing whether or not they think that my research is heading in a good direction, or if it's something that they think is particularly worthwhile. So yeah, it's very important to me.